Hello everyone, my name is Milton Y Nishiyama from Laboratory of Applied Toxicology from Butantan Institute in Brazil, and I'd like to thank ISMB for the opportunity to talk about my work today, prediction and association of alternative splice events in breast cancer subtypes based on meta-analysis approach developed by my student Nathan de Oliveira Nunes. Breast cancer is one of the most diagnosed cancers among women in the world, and it can be classified mainly in 4 subtypes that you can see in this picture that has all these characteristics. The subtypes are the luminal A, luminal B, HER2, and triple negative. That's negative for the first 3 subtypes. So the triple negative is one of the most studied and important because it's the most aggressive and lethal molecular subtype with the worst prognosis. Based on that, we have used the RNA-seq expression profile data from GDC portal for breast cancer. The RNA-seq data analysis, we have developed our own approach summarizing this framework that can be organized in 3 main steps: the classification of breast cancer subtypes, the library size normalization, and a cross-sample normalization, followed by meta-analysis for the prediction of responsive genes and pathways for each breast cancer subtype. Based on the classification of subtypes, we select a subset of the NBC samples to the prediction of alternative splice events. So, based on the around 3,000 RNA-seq breast cancer samples from GDC, we have applied 2 approaches, the AIMS and our in-house approach for breast cancer subtype classification. In the end, we have produced in this table this total number of samples for each one of subtypes, including the normal samples. So, with these classified samples, we can see in the left side the box plot with a higher variation of expression profile between the samples. And in this heat map of experimental correlation, we cannot see a good separation for each sample. So, based on that, we have applied 2 approaches of normalization. The first one of library size with TMM, and the second one with a cross-sample normalization with combat. So we can see in the right side the box plot with a very improved variation between the samples, and in the heat map for the experimental correlation we can find very good sets of samples with very different and exclusive expression profile that could be possible to classify the subtypes. So, based on that, I will explain about the alternative approach for the breast cancer subtype classification, where we select the highly expressed genes for each sample based on the quantile of 5% ranked expression profiles, filtering for genes with the highest standard deviation between all samples, and concluding with the hierarchical clustering approach to classify the sample subtypes and respective genes. So based on that, in this picture we can see the classification and comparison between the AIMS and our approach. And for the TNBC samples, we can see in this column orange box a very good separation, and in this orange box in the line we can see probably a very good set of genes related to TNBC. So after that we realized the analysis of meta-analysis for all the samples for the identification of significant genes and pathways based on the rank product approach. So for the significant genes, we crossed then to the canonical TNBC pathways and the reactome pathways, and we show here in the Venn diagram on the left the significant genes for each subtype, and we can see the exclusive and common genes between them. So in the right side, their respective canonical TNBC pathways, we can see the exclusive ones and common ones, where we can see that the most pathways are containing in basal-like in TNBC. That shows that our canonical pathways for TNBC are really related to TNBC. So, following our alternative splicing prediction analysis, we have used two approaches of string time cuff links to the prediction of alternative splice genes and events, and identification of differentially expressed genes and alternative splice genes for TNBC compared to normal samples. So in summary, we can see a comparison between the two approaches, string tie and cuff links, about the enrichment events for the prediction of alternative splicing. So in summary, we can see that there is a significant loss of intron retention in cancer for the string tie approach, a significant loss of alternative transcription start site in cancer for the cuff link approach, and a significant gain of alternative 5-line acceptor site in cancer for both approaches. So both results are pretty similar. And summarizing these results, I'll show the gene IFNR2 that were presented differentially expressed for both approaches, presented equally 6 alternative splicing isoform events, with most significant loss of exons in their isoforms. So in summary, we can see that the gene and sample normalization approaches are very effective and efficient. The in-house and Ames approach for subtypes classification and remove outlier samples were very efficient. The meta-analysis approach could identify around 8% of canonical TNBC pathways and exclusive ones. The alternative splicing events were in cancer show a very dysregulated profile that may play an important role in that disease. And there were predicted around 600 differentially expressed alternative splice genes for both approaches, with 37 common ones. Thank you very much.